All right, guys, just keep on coming in. We'll just give one more minute to get people going here. All right, if I can get some of you just to do a quick type if you can hear me, a yes, well, a yes if you can hear me, so I know in the chat. All right, awesome. All right, well, I hope everyone had a wonderful spring break. Even though we can't do anything or go anywhere or anything like that. Highlight is to go on a shopping trip to get groceries, but that's all right. Okay. Oh, we still got a few coming in here. Okay, perfect. Well, people can slowly make their way in. All right, so a couple of things I wanted to talk to you about. So first of all, your molecular biology unit quiz is open and it is on the Moodle website. So when you have an opportunity, you want to get that done. All right. So what I want to do is just show you a couple of things. So the first thing I want to show you, I sent you a link on Saturday morning about the calendar for this class. So I just want to show you how that works. So I'm going to share my calendar with you. So here we are, like so. All right. So when you click on the link that I gave you for the calendar, there we are. When we click on the link that I gave you for the calendar, that's going to take you to this page. Now you can't click on the link on your phone. You have to do this on a desktop. Okay, so we played with this a little bit to make sure this worked. So go to your desktop, you click on the link, and then it asks if you want to accept the calendar or not. So you're going to say yes, you want to accept the calendar. So what does that mean? So if you take a look at the bottom right-hand side of my screen, so this is my Google Calendar, and I'm going to just scroll it down here. And on the bottom left-hand side, what we see, and I'll just highlight this here. So what we see here is all of your calendars. Okay, so here's a whole bunch of different calendars. The link I sent you is for Bio30. So that would then, once you've accepted the calendar, you would see it in this link. And then what you're going to do with that is you're going to click on the Bio30. And then what that'll show you is all of these wonderful little events that are going on with Biology 30. Okay. So there's all the events that are happening with it. And if you click on a certain event, so for example, if I click on the biology quiz, okay, so my molecular biology quiz is open. If I click on that, there's the link to it on Moodle. So I can click on that link. It'll still ask you for your username, password. So you'd enter those that you do normally for Moodle, and it'll take you right there, okay? My office hours from 10 to 12. If you click on that, there's the office hour link to Zoom. So you can just go straight to Zoom that way. So what this does is it's just a way to help you get yourself a little bit organized as far as when everything's happening. The assignments, so I just posted an assignment for Hardy Weinberg, okay? The assignments are automatically uploaded, okay? Well, I'm hoping that this will be helpful for you guys to keep organized, and I'm certain you could probably talk to your other teachers about sharing their calendars with you as well. You can go to Calendar in Google Classroom, and what I was hoping is all these events I could upload into Google Classroom, but there's no way to do that. I did a whole bunch of research on that. But what I can do is share this with you, and then all the Google Classroom dates get uploaded onto this. So hopefully this is something that helps for you. So like I said, you have to do this on your desktop. Once you've added it, so once you've added Bio30 on your desktop, then you can go to your phone and open up Google Calendar on your phone. You have a similar list of my calendars. All you have to do is check off Bio30, and now you have it on your phone all the time. Okay? So you only just have to set it up on your desktop, and then once it's set up, then you're good to go to your phone and use it. Okay? So yeah, so I'm hoping that that's helpful for you guys, because I realize that you're getting a million messages from all your teachers, and I'm hoping this is one way to kind of help alleviate some of that stress. Okay, so that is what that's all about. You had a DNA protein synthesis assignment, so if I click on that, that was due today at 1, so I'm hoping that everyone has that in. For that, I haven't looked at Google Classroom to see. I did share a link with you guys on the online tools of Google Classroom on how to link, if you're having a hard time linking assignments, to turn them in through Google Classroom. So there is a link there for you. But again, if that's too difficult, isn't working, just send me an email with your assignment or share it with me through your G Drive. Okay, so that's enough of that. The other things I wanted to share with you. So remember, your molecular biology quiz is open, and we're going to look at population genetics. So population genetics, so I'm just going to share my PowerPoint with you. So here we are. I'll just scroll through to where we were. So I didn't go through all of these examples, but the examples were there, and I had put in my notes on the Google Classroom stream to go through these on your own and then do the practice sheet. So on the practice sheet, I gave you answers. So I posted answers there in my population genetics folder on Google Classroom with classwork. Oh, just one second here. It's so question about the quiz. It's molecular biology, so it includes all of the DNA and protein synthesis stuff on the quiz. So before you sit down to write the quiz, you want to have a calculator in front of you and your data sheet in front of you, because you're going to need both of those. Okay. What else was I going to say? Oh, so I did share all my answers. I did them in an online format, and I made a video. So it's a YouTube video that's linked in the population genetics folder that answers those 5 questions that I gave you as a practice assignment. Okay. All right. So we're going to move on. And I just wanted to do a few more practice questions together. So this link here that's in your notes, let me just open this up here. Slideshow, current slide. This link that's in your notes, we're not going to go to that link. I've already cut and pasted these questions into a different format. So I'm going to share this with you, and we are going to work through these questions together. Just find out where I put it. There we are. All right. So what we have is, let's get rid of that. Can you all see this screen? I just want to make sure that it's sharing properly. Can you all see a bunch of little pigs on the screen? Okay, perfect. All right. So let me just get all my stuff out of the way here. And move that over to the side. And get rid of this. Okay, so the question is, the pink coat trait is dominant over the black coat trait. We want to determine the frequency of pigs that are heterozygous for coat color. Now, typically frequency is given in a decimal format, okay? So I want to figure out the frequency of pigs that are heterozygous, okay? So I know that heterozygous pigs, that formula for heterozygous is 2pq, okay? But the problem is I don't have a P and I don't have a Q. Now, when I'm doing these problems, like it says in your notes, I either have to start my problem with an allele. Okay, so my problem's either going to start with an allele frequency, or my problem's going to start with a Q squared. Okay, those are the only 2 ways these problems can start. So either an allele frequency or a Q squared? Okay, so any suggestions? So using the chat there, any suggestions with how I'm going to figure this out? What would be my first step? Okay, in order to calculate heterozygous, 2 times P times Q, I need a P or a Q. So what am I going to do? Okay, I know pink coat is dominant over black coat. Okay, let's take a step back. What does Q squared stand for? Don't all answer at once. Yes, Q squared stands for the frequency of individuals. This is really hard to write on. Frequency of individuals that are homozygous recessive. So when Eric writes here the frequency of the recessive trait, absolutely, right? So is there a way, so I don't have an allele frequency, that's not given to me in the question, but this is given to me indirectly. Right, so what is my frequency of black pigs? How am I going to figure that out? What's my frequency of black pigs? Okay, I'm looking at my pig population. What's my frequency of black pigs? Because that's my recessive trait. You might have to do some counting. 4 out of 16, yes. Okay, so there's 16 pigs in total. It might be hard to see depending on the screen you're looking at. So there's 16 pigs in total. 4 of them are black. Okay, exactly, guys. So let's take a look at that. So I have Q squared equals, because that's my frequency of individuals with a recessive trait, Q squared is 4 out of a total number of 16, which equals 0.25. Now, that's Q squared, but I'm not quite done yet because I need to figure out 2 times P times Q, right? That's what I need to know. So what's my next step? What am I going to do next in order to get to this point where I have a P value and a Q value? Awesome. I need to square root the 0.25. Okay, so I've got to get a Q value. So I know that Q is equal to the square root of Q squared, right? 0.25. So Q is equal to 0.5. All right, so we've got a Q value. Okay, that's good, but I have 2 times P times Q. I need a P value. So how am I going to figure out P? Exactly. I know that P plus Q equals 1, right? So I know that P is equal to 1 minus Q, which is equal to 1 minus 0.5, which equals 0.5. Okay. So I have a Q value, I have a P value. So now I'm looking for the frequency of individuals that are heterozygotes, right? That have big A, little a. So I know 12 of my pigs are pink, but I don't know what the frequency is of individuals that are heterozygous, right? So I go like this. I know that my formula for the frequency of heterozygotes is 2 times P times Q. So 2 times 0.5 times 0.5. And what do I get? Exactly. So if I wanted to do this in decimal form, I would just say, and usually they ask you for two numbers after the decimal. So 0.50. So half of the pigs in that population are heterozygous. So 8 of the 16 pigs are heterozygous. All right, any questions about that problem? So what I like about this question is we have to get information from looking at a picture, right? And then we have to figure out frequency by counting. So I do like that style. So that's why I like to ask this particular one. Okay, we're going to do another one. So I'm going to move forward to my next slide here on my smart notes here. Okay. So let's read through what this is asking us. In a certain population of 1,000 fruit flies, 640 have red eyes, while the remainder have sepia eyes. The sepia eye trait is recessive to red eyes. Determine the number of individuals expected to be homozygous for red eye color. Okay, so we need to know the number of individuals, but we can't figure that out until we get frequency. So my first step is to figure out the frequency, or my step is to figure out the frequency of individuals that are homozygous for red eye color, which is 2 big A's. What formula do I use for that? Awesome. Okay, so my formula for that is p squared. Good job, you guys. Okay, so my formula is p squared. All right, well, my problem is I don't have a p, I don't have a q, so I need p in order to figure that out. But I have to remember that these questions always start in one of two ways. They either start from giving me allele frequencies, or they start from giving me a q squared. So what do I get with this problem? Okay, awesome. Thank you, Ashley. I cannot. So Ashley did great work there if you're looking at the chat. So I just want to take that one step back and show what sometimes is done incorrectly. So don't copy what I'm going to write right now because this is going to be wrong. What some people do incorrectly for these is they look at it and say, okay, we've got 640 that have the dominant trait. So I'm going to say that p squared, don't know why my 2's aren't writing very well here. P squared equals 640 over 1,000. But that's not right because p squared is only the individuals that are homozygous dominant. This 640 is individuals that are homozygous dominant and individuals that are heterozygous. So if I wanted to write this properly using the dominant trait, I would have to say p squared plus 2pq equals 640 over 1,000. Well, that is completely useless to me because now I have a p as a variable and a q as a variable, and I don't have another equation to try to solve systems, and in Bio30 we'd never do a systems equation anyway. Okay, so looking at the work that Ashley did, what we have to do is we have to figure out our q. So I'm going to get rid of all this stuff that I just did because it's all basically wrong. Okay, and let's go back to this. If 640 have red eyes, then we know that 360 have to have the sepia colored eyes because that's the recessive trait. So I'm looking at the frequency of individuals with the recessive trait is 360 over 1,000, which equals 0.36. All right, there's my q squared. Now I need a p, so what's my next step to get the p value? Awesome. Okay, so Q, my next step is to figure out what Q is. Q is equal to, okay, so we're dividing 360 by 1,000 because remember I'm looking at a frequency. What's the percentage of them? So I can't just say 360 because that's a number. I need the frequency. So 360 divided by 1,000 tells me what percentage is in that population. Okay. All right, so Q equals 0.36, the square root of 0.36, which is 0.6. Okay, Next step. Awesome. So now we're going to get P. So P is equal to 1 minus Q, which equals 1 minus 0.6. So P equals 0.4. Okay. So I'm looking for the frequency of individuals that are homozygous dominant. So my formula for that is P squared. So now I have a value for P, so I'm going to square that. And what do I get? Awesome, good. So I'm just going to move this up to the top here so I can extend my page. Okay, so P squared equals 0.16. Now, we're not quite done because the question didn't ask me for the frequency of individuals that are homozygous dominant. It asked me for the number of individuals. So I know that 16% of the population are homozygous dominant. What do I need to do with that to figure out the number of individuals that are homozygous dominant? I'm going to take my 0.16 and I'm going to multiply that by my population size of 1,000. And I get 160 flies. Okay, so 160 flies are homozygous dominant. Okay. All right, any questions about how we did that? So what I like about this particular question is it focuses us on remembering we have to go to that recessive trait. So even though the question gave me information on the dominant trait, I have to use that to find the frequency of the recessive trait, and then I can go on. Okay? All right. I have one last one for us to look at. So this last one is about phenylketonuria, so PKU. So this is a disorder, results in mental retardation if it's untreated in infancy. So every kid, you guys all got a little poke in your heel to test if you had this or not. Okay, so in the US, 1 out of 10,000 babies is born with this disorder. Calculate the frequency of individuals that are carriers, so therefore heterozygous. Express your answer as a percent rounded to the nearest whole number. All right. So remember how we start these questions. We either start by the question giving us an allele frequency or the question giving us the frequency of individuals that are recessive, that have the recessive trait rather. So this question is asking me for the frequency of individuals that are carriers. So what's the formula for that? Awesome. Okay, so my formula for that is 2pq. So I need to solve this. I need to figure out what my p-value is and what my q-value is. So any ideas what my starting point is? Awesome. Good. So I can see, because they tell me that people have this disorder. They have the recessive trait, 1 out of 10,000. So Q squared is equal to 1 out of 10,000. Okay, next step. Perfect. So Q is equal to the square root of, and yeah, it's easier for me to write this as a decimal, which equals 0.01. Okay, well, I've got a Q value, but I'm still stuck figuring out my P value. So how do I get P here? Okay, good. So P is equal to 1 minus q, which is equal to 1 minus 0.01. So that equals, oh, scribbling some stuff here. Let's go back to my pen. There we are. Okay, so P equals 1 minus Q, so P equals 0.99. Okay. All right, so I've got my P and my Q. What's my next step? Good. So I need to figure out the frequency of individuals that are heterozygotes. So 2 times P times Q, 2 times 0.99 times 0.01. All right, what do I get? Okay, 0.0198. And in these instructions I told you just to write it as a percent to the nearest whole number. So what would this be as a percent to the nearest whole number? Perfect. So this tells me in this situation with this population, 2% of the individuals would be heterozygous or carriers of the PKU trait. All right, any questions about this? Awesome. Okay, so we're going to go back to the notes and we'll see how far we can get. Like I said, I just posted a worksheet for you guys that'll be due to me on Wednesday on this stuff. Okay, mostly on the calculations part and not really on the theory, but you do have to know the theory as well. All right, so let's go back to that. And this. Okay, so back to this slide here, and we're just going to go forward. Okay, so what we have here, evolutionary change occurs. So remember, we know if a change is occurring in a population when those allele frequencies are changing for a particular trait. When the allele frequencies are staying the same for a particular trait over generation after generation, then we know that it's not an evolutionary change, okay? So for example, if we were to examine the tongue rolling, okay, the can versus can't roll your tongue alleles. Well, those, because they fit with the Hardy-Weinberg conditions, we're probably not going to really see a significant change in those, okay? So we're just going to look at these conditions. So one of these conditions is mutations. And I know we talked about this earlier, but we're just going to talk about it one more time. So mutations we have, basically evolutionary change occurs as mutations lead to new alleles being added to the gene pool. So instead of having a 0.7 and a 0.3 for my dominant allele and recessive allele, I maybe have something else that's a 0.1. So now I have maybe a 0.55 and a point, whatever my numbers were, and that's all going to change. Okay. All right. Genetic drift. So this is the example I was talking about with the dog peeing on the flowers, right? And the dog peed on the flowers not because it wanted to pee on that particular flower, but it just peed. Okay, so random changes. So that's important to understand here. Whoops. We're looking at random changes in gene frequency. Here's what the problem is with that. Is if we have these changes, we can have a population that's afflicted with something that's referred to as homozygosity. Okay, homozygosity. Okay, and what that means is the population could become completely homozygous for one allele and the other allele could be gone. So instead of having a big A and a little a, we just have little a's. So every individual would have the recessive version of that trait. Or instead of having any little a's, the whole population just has big A's. So now every individual has only the dominant trait, dominant version of that trait. Okay, so this is what the problem is with this, is homozygosity can happen. And if it happens in more than one of those gene pairs in the body, that can lead to a big change in the diversity. Now, genetic drift has two subcategories that you're responsible for knowing. Okay, the first of those alleles. Hold on. There we are. Oh, whoops. I have just a couple more things to add here. So small populations are much more susceptible to genetic drift, chance events happening in a small population, as we saw with the dog peeing example. Okay. But when we're looking at this, we're talking about the gene being neutral. So that gene, that version of the gene doesn't have a selective advantage or a selective disadvantage. Okay. Otherwise, it's not genetic drift. Okay, so this is one category here. So this is our genetic drift. This isn't one of our subcategories. So what we're looking at with this example is we're starting off with this frequency where our dominant and recessive allele we see is 0.7 and 0.3. Okay. Now, what we're looking at is some plants, only 5 plants leave offspring. Okay. So not very many plants leave offspring. So if I look at this, I'm looking and I'm not seeing, right, my dominant allele and my recessive allele, I'm seeing those frequencies are changing. And now, so only these 5 plants left offspring. Okay. Well, now only these 2 plants. So for whatever reason, only those 2 plants left offspring. Maybe the rest of them got peed on. I don't know. But what happened as a result of that is this whole entire population only has big A's. So 100% of that allele frequency is for the dominant allele, and there's no recessive alleles left. So now it doesn't matter which individual breeds with which individual. You're never going to get a white flower in this population. Okay, so that's genetic drift. Okay, so our subcategories of this. So the first subcategory is bottleneck effect. So the bottleneck effect arises from some kind of catastrophic event happening to a population. Usually when we think about catastrophic event, we're thinking something that we did, right? So basically hunting some species to near extinction. So different textbooks have different examples of this. One example of this is our elephant seals. So we had thousands and thousands and thousands of them, and then we hunted them until we had like 20 left. Okay. Well, let's think about what that impact is. Okay, so disastrous event, population suddenly is just reduced in size. We've done this to our sea otters. We've done this to other creatures, right? So it's suddenly reduced in size. So as it's reduced in size, when I think about that population, right, only a few alleles are coming out of that bottle. Okay, so they're going through the bottleneck. And here's my surviving individuals. So if we think about the gene pairs, right? So let's say in our original population, we had big A's and little A's and big B's and little B's and big C's and little C's. So we're thinking about like 3 different possible gene pairs. We know that there's going to be thousands of them, right? But with the individuals that survived, let's say the only individuals that survived were recessive for the A trait. And maybe we're all homozygous dominant for the B trait. And maybe we're recessive for the C trait. Well, what that means is even though the population has now had an opportunity to grow, which has happened with the elephant seals and the sea otters, now that we have hunting restrictions, those populations have been able to build. But the thing is, no matter how much they build, we're never going to get a big C, a big A, or a little B. And that's homozygosity. What they've noticed in studies with the cheetahs is there's a lot of gene pairs that are homozygous with cheetahs. Now, no one knows exactly what happened, but some catastrophic event likely happened with the cheetahs to lead to so much homozygosity, which means the difference between one cheetah and the next is minute. Okay, they're very closely related to one another, which isn't good when we talk about the importance of genetic diversity. Okay, so that's what the bottleneck effect is. The other sub-example of genetic drift, let's get to this here. Oh, whoops, another bullet here. As this population increases, the gene pool will still be small. It'll lack diversity, okay, as we just said. Okay, so founder effect. So what's happening with the founder effect? So the first thing I want to just write up here is this is not this is not immigration. Okay. So what I mean by that is these individuals are not joining a pre-established colony. They are creating a brand new colony that never existed before. Okay. So what's going on? I'm just gonna forward this so we can see my bullets. Individuals leave the original population to form their own population. Okay, so they're not forming or adding to in a pre-existing, they're forming their own brand new population. The allele frequencies will be different. Now, we can have homozygosity occur here as well. So when I look, and it's really hard to see all the little gray dots, we have blackish dots and we have gray dots. So when I look at these frequencies here, I've got these little pink ones. We can't see any colors, but light gray. So we have a 2-to-1 ratio. Whoops. We see a 2-to-1 ratio here of the dark to light. Okay. But now this little tiny population of 4 individuals decides they're going to build their whole new population. The problem is, whatever frequencies are here, those are the frequencies we're going to see in our new population, right? We're not going to see the frequencies of the original population. So one of the prime examples of this is with the Dutch Amish in Pennsylvania. So this group of individuals moved from the Netherlands to the Pennsylvania region. Now, this makes for a good study because this individual is fairly this population is fairly isolated, so we don't have a lot of emigration as far as the genes go. But what happened is in the regular population, there was a particular recessive trait that caused short limbs, okay, short arms, short legs. And in the regular population, so in this original population, that frequency was pretty small. But as they moved here, because one of those individuals had the recessive trait, that trait was at a very much higher frequency in the new founding population. Okay. And that's as a result of the founder effect. Okay, so any questions about bottleneck effect or founder effect? Okay. All right, so we'll move on here. Okay, so the next one we have is migration. So migration is talking about immigration and emigration, okay? And the fancy bio term when we talk about that is gene flow. So we want to imagine genes are flowing from one region to another region, and that's why we call that gene flow. So the gene pool of a population changes depending on the movement of individuals of the species, okay? All right, so immigration and emigration, and these are terms you need to know, especially when we get to our next section, but immigration, moving into a population, emigration, it's moving out of, so we can think about that as exiting a population. So my dad immigrated to Canada, he emigrated from Denmark. Okay, so we want to make sure we're clear on what those terms mean. Okay. How much the migration actually affects our evolutionary change really depends on the genetic differences of the individuals either coming in or leaving, right? If there's a small group that's leaving that has a particular trait that a lot of other individuals don't, then we could see a big change in that trait, right? So it really depends on what the differences are in the genetics, so genetic differences, and how quickly that's happening, right? All right, our next example or next. Okay, so here we're looking at gene flow. So we have our western deer population. Oh, sorry about that, guys. We have our western deer population and we have our eastern deer population. Okay, So there's our western deers, there's our eastern deers. Oh, but some western deers decided to join. There was a pass that they got through and they joined that population. Okay. So a couple of things that we need to talk about with this example here. First of all, what has happened to the genetic diversity of the gene pool of the western deer population? Has it increased or decreased? Okay, so I'm talking about the western deer population. Has its gene pool increased or decreased? Yeah, awesome. So the diversity has decreased, right? Because we have some individuals that have left, and who knows what recessive genes they were kind of holding on to, right? Okay, what about the eastern deer population? What's happened to their gene pool? Yeah, it's increased. It's gone up. Because remember, the gene pool is just all of those alleles of all the individuals in that population. And again, one of the misconceptions is that these new individuals have to make babies before those genes become part of the population. That's not true. They just have to walk through the door. As soon as they join that population, their genes are part of that gene pool. And they have now increased the gene pool of that population. Okay? All right. Natural selection, right? The gene pool of a population changes when there's a genetic advantage or disadvantage, right? We've talked about this already. Disadvantage, a selective disadvantage. So if a particular trait caused by one version of that gene causes an individual to not be able to reproduce, to pass that on, that's considered a disadvantage. Okay. All right. And if there's a disadvantage, we should see the frequency go down over time in the population. All right. Non-random mating. So individuals choosing, so non-random. Random mating means don't care. Non-random means that we're choosing a mate because of a particular trait. Now, again, when we're thinking about the Hardy-Weinberg principle, we're looking only at a particular gene pair. So am I choosing a mate because they can roll their tongue versus not roll their tongue? Am I choosing a mate because they have a beautiful long tail versus they don't? Am I choosing a mate because they have a beautiful dance ritual or their dance ritual sort of sucks, right? So that's what I'm looking at. So if a mate has traits or characteristics that make it so that they're more likely to breed, then that's going to be an advantage. Okay, we're going to see that more often. And that's where we get the peacock with the big long tail. Okay, so, all right. This here, I would just try this link yourself. It goes through the 5 Hardy-Weinberg conditions that you can take a look at. All right, so coming up for us, you have a molecular biology quiz. It's open until 8:30 on Wednesday. You have a population genetics worksheet that's due to me. So I shared that with you on Classroom, and that's due to me Wednesday at noon. So make sure you get that done. And we're back here Wednesday at 2. All right. Any questions before we sign off? Okay, looks like I don't have any questions. Well, I hope you guys are doing good. You might want to go for a walk today. It actually looks like the sun is shining. So get your work done, guys, and we will talk to you later. All right. Bye-bye. You're welcome, everybody.